So you might have heard the saying, prevention is better than cure. This can be literally applied to a groundbreaking technology called CRISPR, which gives humans the potential to prevent a disease before birth. Let's start at the beginning with DNA. This stuff codes for every living thing on the planet. There are more than 3 billion base pairs in the human genome alone. Now, let me introduce you to the war between bacteria and viruses. They've been fighting for at least 1 billion years. Here's the strange part. Scientists found that some DNA in bacteria matched with DNA from viruses. Believe it or not, this observation is the foundation for the CRISPR breakthrough. The star of the show is DNA's less popular counterpart, RNA. Just like when taking a photo, the CRISPR system saves a snapshot of a defeated virus's DNA. This allows CRISPR to recognize and defeat the virus later on. And remember the CRISPR acronym? It refers to repeated DNA sequences that neatly separate the viral DNA. They're like genetic filing tabs. The CRISPR system we're talking about here employs 3 helpers: tracer RNA, cRNA, and an enzyme. In this case, we'll say the Cas9, or CRISPR-associated, enzyme is being used. This entire defense system moseys up to the intruder, and the cRNA gets to work looking for a match. If found, the enzyme cuts the virus's DNA and disarms the attacker. But how do ancient bacterial defense mechanisms relate to us? To answer this question, we need to understand what makes CRISPR technology unique from other gene editing methods, which are, in fact, not new. CRISPR editing is unique because scientists can specifically control which genes are targeted. This could have huge implications, some more practical than others, like eliminating antibiotic resistance, engineering more robust crops and livestock, treating diseases like cancer, and helping to alleviate pandemics such as that of COVID-19. But natural systems aren't always perfect. Being a new discovery, there may be unknown consequences of CRISPR editing that haven't been identified. And like most systems, CRISPR could make an error. When applied to humans, this brings up the issue of erasing bad genes, which is a debate in and of itself. Who gets to decide which differences are eradicated from our DNA? This concern brings new meaning to prevention versus cure. The main takeaway here is whether we as a species are willing to take the next step in history by editing life itself. Indeed, this massive potential brings up ethical concerns, making CRISPR's usage the moral conundrum of our time.